a pleasure to be here talking to you all this morning or afternoon, as the case may be, about sweetness and the biological drive for sweet taste. So, I think it's important to really go back and think about, why do we taste? What is the role of this sense? And we know that every species has to solve the fundamental problem of obtaining sufficient nutrients and avoiding being poisoned. A species' taste sensitivity and preference is co-adapted to its ecological niche. For instance, we know that the obligate carnivore, the cat, does not have a functional receptor for sweet taste. But in fact, the genes for sweet taste receptors evolved over 400 million years ago, before the first vertebrate even walked on land. And you can see this image here of what that creature might have looked like that first evolved this ability to detect sweet compounds. So this sense really evolved as a nutrient signal, so that we can find the nutrients that we need in our environment. So what's important to understand is that in order to recognize these nutrients and to remember that this is where we can get nutritional value, the senses of taste and smell are the basis for flavor-based learning. So taste and smell are the cues that really enable us to learn about the health consequences of the things that we consume. Are these things going to be toxic? And often bitterness is a signal for toxicity. Or are they going to be nutritionally valuable? Things like sweet or sugar or fat or protein. And so we have positive or negative hedonic responses, and these responses shape our behavior in order to ensure that our nutritional needs are met. Sweetness in particular is a signal for the presence of calories. And sweet liking, it's very clear, is present at birth in all cultures and even in primates, other primate species. So really, you can understand why it's so difficult to redirect the effects of this survival-driven biology and learning. And in fact, this has been attempted because we knew that with salt preference, if you go on a diet that's low in salt for some period of time, your ideal or your optimal preferred concentration of salt does decrease. So you can actually shift your preference. So can we shift preference for sweetness? And this is one small study that was done. There's two larger studies currently in progress to looking at this question in more depth, and it'll be exciting to see the data coming out from those studies. But for the moment, this is really the only study that addressed this by Paul Wise and colleagues, where they reduced sugar intake by 40% for 3 months. And they looked at preference for a couple of different foods, and I'm showing here the results for the sweet pudding. And what you can see is that really there was no change in the perception of intensity over the first 2 months of the lower sugar diet, but the intensity was perceived as more intense for the low sugar pudding after 3 months of this low sugar diet. If you look, they did then at this point they went back on their normal diet, and you can see that intensity started to come back towards where it was before being on the diet. So it was a temporary effect. The other thing, in addition to looking at whether the subjects perceived the sweetness as more intense, they asked about their liking. And unfortunately, there was no effect on the preferred level of sugar in the pudding. So even though at this point they perceived the sweetness as more intense, when they tested their preference for pudding with different levels of sugar, they just preferred the one that was more intense, that was the same concentration as in the beginning. So it's clearly very hard to shift this preference. However, the studies that are in progress are looking more comprehensively at things like equating the amount of sweetness in the diets of these individuals. In this particular study, it was not a fully controlled dietary study where they knew all of the foods that the individuals were eating. So it was more of a pilot study. So to start understanding how we can attack this problem, we really need to think about how sweet taste works. And the oral taste cells are the initial chemosensors of the alimentary tract. So, you know, they are present in the tongue, but they're also present throughout the gastrointestinal tract, and that's an important feature as well. But if we look at the tongue and up closer, we have these taste papilla, and within the grooves here are the taste buds. And the taste buds hold these taste cells, and there's multiple types of taste cells. And it's very important to understand as we think about modifying taste, modifying sweet taste, that anything that is tasted has to go into this pore, which is about the diameter of a human hair, and it has to be dissolved in saliva and the mucus that is present in this taste pore. And we have different types of taste cells. We have cells that sense salt or sour or sweet or bitter. By and large, these taste qualities are sensed by different cells, different cell types. The other interesting thing is that taste cells are replaced about every 10 days. And so this provides the opportunity for the system to be shaped by experience. And we do know that there are changes in the density of different types of receptors, for instance, bitter receptors in particular, with experience. So if you are exposed to a high level of particular bitter compound, the amount of that receptor is downregulated. So this helps the system to be responsive to its environment. So as I said, for the most part, the cells are single modality, but we have learned that crosstalk can occur between these cells. And also the neurotransmitter for taste is the chemical ATP, which is the cellular form of energy. So when a cell is activated, that triggers a cascade of events within the cell, ultimately triggering the release of this ATP, which signals the taste nerves. Zooming in farther, I just wanted to make a couple of points about the receptor itself. So this is the sweet receptor. It's two 7-transmembrane domain helix receptors that form sort of a clamshell shape on the outside. But it's very interesting because it has a lot of different sites that different types of sweeteners interact with. So sugars, aspartame, some small molecule sweeteners interact with the outer part of the receptor. But there's other types of sweeteners like brazine or cyclamate that interact with parts of the receptor that are more into the membrane. So the cell membrane would go through here. And what that means is that these different types of sweeteners will have different kinetics or different onset and offset parameters. So the duration of the sweetness, the rapidity with which the peak sweetness occurs, and so forth, will all vary. And part of the reason for that is because they're binding to different places on the receptor. And it's a lot harder for something that's binding down here. It's going to take longer to probably trigger the sweetness, and it's going to take longer to decay afterwards. And I think, as you'll hear later, one of the concerns that people have is that these other alternative sweeteners don't have the same onset, peak, and offset that regular sugar does. So, we're not very good at fooling Mother Nature. We do know that our brain can distinguish caloric from non-caloric sweetener, even when the sensory experience is matched as best we can. And reward areas are differentially activated for different types of sweeteners. But this tells us that we know they're different, but it doesn't really tell us why. So to understand that why, it will help us to craft foods and diets that can satisfy our sweet tooth with less sugar and help to maybe devise ways to shape our preferences to accept less sweetness in our diet.